So Dr. E pretty much opens this PowerPoint with the survey question, and it's consider how transcription. So first of all, transcription, what is that telling you? What are we doing? Transcription.
That's from, oh my gosh, DNA to RNA.
Correct. So consider how transcription initiation is affected when the RNA polymerase cannot bind properly to. That's the hint. The question is, which of the following is most likely outcome of this mutation? And the mutation is altering RNA polymerase's ability to recognize the promoter region. So the promoter region of the gene, where is it located? There's like a specific term that scientists sometimes use. It's upstream. It's just basically
upstream?
Yeah, it's
okay.
So get a dry erase marker. So if this is the gene, this is 5 prime, this is 3 prime, then this is upstream.
Okay.
So in front of it, this is downstream. So promoter would be right here before. Upstream, yeah.
Okay.
And the promoter region is it's a sequence of nucleotides that allows for RNA polymerase to bind to it and then transcribe the gene. So what would happen if there was a mutation in the promoter region?
There would be less transcription.
Correct. Yes. So for this specific, like you see right now, you did not know the multiple choice questions.
Yeah.
But the correct answer, the first 2 words are reduced transcription. So just you would be able to reason this out even without you looking at the questions, and that's just like a good sign to be in this position in order to avoid that blacking out because of being nervous.
Yeah.
Okay, so the next question is, so the little passage that he gives is, a medical student is examining a case where a patient has an intron retention disorder leading to improperly spliced mRNAs. They must determine the downstream effects on protein synthesis and cellular function. So the question is, what is the most likely consequence of intron retention in mRNAs? So we are keeping introns. Are introns supposed to be kept?
No.
No. Introns are like the intruders, so they're supposed to be cut out. So for this, I'm gonna give you the options because there's for this question, if it was an open response question, you could say multiple things and be correct.
Yeah.
So tell me, in your opinion, what could be one of the things that could result from intron retention?
Well, it's almost like the mutation, right? It would lose.
Yes.
But it wouldn't be in that transcription part. It would be.
So I mean, the transcription is going to be affected, but it's not the main cause of. It's like intron retention causes transcription to be affected, but because they just follow each other, so it's like a secondary effect. But yeah, so the protein that's gonna be made, it can be longer or it can be shorter, pretty much.
Okay.
So introns, if they are kept, and for example, if there is a stop codon, so there's 3 types of stop codons.
Yeah.
It's U. They all start with the U.
Mm-hmm.
UAG, UGA, and U. UAG.
It's okay.
Codons. UAA. Okay, I thought I was UAA. So, the way you remember, because I think this might be a multiple choice question.
Okay.
Like stop codons, it's like something bad, right?
Yeah.
I mean, not something bad. It's good, but like think stop. So it's U R grows, UAG.
Oh, okay.
You go away, UGA. You go away. You are annoying.
Oh, okay.
So, and then if one of the introns that was kept has any one of these, then as soon as RNA polymerase runs into these, it's gonna stop transcription. Yeah. So the mRNA is gonna be much shorter than it needs to be. It's gonna be missing needed parts. But it's also possible. It's highly unlikely. But it's also possible that all of those introns, none of them are gonna have a single stop codon. So instead of stopping early, the resulting mRNA, the RNA polymerase is gonna stop at the same stop codon as it's supposed to do, but it's just gonna transcribe a much longer piece of mRNA, which is gonna translate in the ribosome into a completely different protein, and that protein is gonna become the non-functional. So intron retention, in essence, leads to non-functioning proteins. And the question right here is, what is the most likely consequence of intron retention? And the correct answer is
B, right?
loss of translation initiation and truncated proteins. Yes.
Yeah, because since it affects it, then it wouldn't increase protein. It wouldn't enhance the stability of the mRNA, and it says no effect, but there is an effect.
Yes, correct. Exactly. Perfect. Like that's the type of reasoning that you need to bring with yourself to the test. Okay, so this slide is pretty much central dogma is just DNA to RNA, right? But viruses are exception. Okay, we don't get a lot into viruses, but just know this is like the central dogma. But again, it does have some exceptions to it. The introns are cut out, exons are kept. mRNA translates into proteins. So the RNA preparation, RNA maturation consists of intron splicing. Then you get the 5' methyl G cap, which is just the cap that allows the mRNA to bind to the ribosome, and then a polyadenine tail, which keeps it stable, keeps the end tail from degrading, because mRNA is an unstable molecule. Okay, I went over this on Thursday.
Yeah.
And I think this is just like general idea about DNA and RNA, but I think it is helpful. So DNA, it's deoxyribose nucleic acid. So deoxyribose nucleic acid. So deoxy means that we're missing an oxygen. Ribose, it is just a type of sugar.
Yeah.
You haven't taken organic chemistry, if I remember correctly, right?
Yeah, I'm taking it now.
You taking it right now?
Yeah.
But you haven't taken. Is it 333 or 334?
333.
333. Okay, so you're not there yet, but sugar molecules, they're made up out of carbons and oxygens and hydrogens usually, and then let's say that they are circles. They are, I mean, they're either hexagons or they're pentagons. For the simplicity, it's a little bit more complicated than that, but
Yeah.
you get a 334. For our purposes, that's all we need. So a ribose, it is a 5-membered ring. And it looks like this.
Yeah.
So this is ribose, but we have deoxyribose. So in DNA, it's forming RNA. In DNA, this right here is just a hydrogen. It's not an oxygen.
Okay.
So you're literally missing an oxygen. So when you say RNA, it's ribonucleic acid. Is that what? Yeah, ribonucleic acid. Because you have all of the oxygen that are usually present in a ribose. And okay, so, and you know how professors love to say that DNA is made up of a sugar phosphate backbone. So I think it's sometimes like hard to understand, put it like in kind of real terms for yourself. And so pretty much how it looks like. So DNA is a double helix, and like usually it's drawn like this, right, or something like that. And then you have these like lines between them. So this is obviously like when this is drawn, this is a artistic representation of DNA. It kind of looks like this, but it's not straight lines at all. It's made out of like molecules. So these right here are hydrogen bonds between nucleotide bases, and then this line right here, in reality, so you have so this is DNA, okay? So what I am trying to show you right here is that it is a repeating system. So you have a phosphate, which a phosphate is a P that has 4 oxygens bound to it. 2 double bonds, 2 single bonds. And then this goes up, obviously this goes down. And so you have oxygen, phosphate, oxygen. And then, so again, we're returning to the 5' to 3', just for you to be able to see why that is. So again, this is organic chemistry stuff, you'll learn it.
Yeah.
But essentially, this carbon is labeled number 1, So it's 1 prime, 2 prime, 3 prime, 4 prime, 5 prime.
Okay.
So like for our purposes, just know that this is 5, this is 3.
Yeah.
So this one right here is 3, this one right here is 5, number 5. We call it 3 prime, which is essentially the same as 3 prime.
Mm-hmm.
And when RNA polymerase moves along the backbone, it's gonna move, always encountering number 5 carbon first, moving to 3, going to phosphate, going to 5, going to 4, going to 3, going to phosphate, going to 5, going to 4, going to 3, going to phosphate. And the nucleotide bases, the A, T, G, and C's, they're connected instead of this oxygen right here. They're connected here.
Okay.
And then you have this same thing arranged anti-parallel. So if I were to draw it, it would be flipped around because we have to have the 5 prime carbon first and then the 3 prime. So it would go this way.
Yep, sorry.
The opposite way.
Okay.
Anti-parallel. So and then RNA would be just obviously one thing. Over here would be OH instead of H. And I think that just presence of that extra oxygen, I'm pretty sure that's what it is, makes it less stable.
Less stable.
There's like more electrons there, more repulsion, more charge. That's why we store our information as DNA, which is like a long-term storage format of it. Okay, so transcription is converting DNA to RNA. It is initiated by RNA polymerase. It requires transcription factors and that binds to the promoter region, and then RNA polymerase binds to the promoter region in the nucleus. And then so the difference between one of the differences between prokaryotes and eukaryotes in their translation transcription type processes is the fact that so our cells have a nucleus, prokaryotes do not. Prokaryotes, RNA polymerase is going to be a star, okay? So it comes, it begins replicating mRNA.
Yeah.
And even before this is done, this can be bound to a ribosome and begin generating proteins. So at the same time, it's being transcribed and translated.
Okay.
We have a nucleus which doesn't allow ribosomes to get in the nucleus. So for us, transcription has to be completely finished and mRNA has to be processed, and only after mRNA has left the nucleus, we're able to begin translation. So I think that could be like a multiple choice question with multiple correct answers, how Dr. E does it, which asks, for example, what are the difference between a prokaryotic and eukaryotic cell? And prokaryotic cells do not have introns or exons, if I believe. Yeah, it says they're rare in prokaryotes. So like for this class, I'm sure it's like that they're non-existent. And then in prokaryotes, transcription and translation of the same mRNA can be happening at the same time because there's no nucleus, and multiple ribosomes can be even doing it at the same time with the same mRNA strand. Obviously, there's no nucleus, there's no organelles. The genome is one singular DNA, it's not chromosome. There are chromosomes. What else? Yeah. Okay. Okay. And then, so also another big thing about RNA and DNA difference is in RNA, thymine is uracil. So T is U.
Okay.
U is T. CAG stay the same, but T becomes U. And now going back to this, these are hydrogen bonds. Hydrogen bonds are bonds between molecules. So within a molecule, there can be covalent or ionic bonds. Ionic bonds are electrons transferring between metal and non-metal. Covalent are usually between two non-metals, and it's the sharing of electrons. These bonds are stronger than hydrogen bonds, but between intermolecular bonds, hydrogen bonds are the strongest. So it's not actually a bond like H2O.
Yeah.
Like this is a covalent bond. And hydrogen bonds, they are just, so you know, in the beginning of 333, who's your teacher by the way?
Millikan?
Oh, perfect. I had him. So he goes over electronegativity, right? Oxygen is one of the highest electronegativity. So electronegativity is how strongly it attracts electrons. Yeah. Electrons are negative in their nature. So this is much more electronegative than hydrogen. So all of the electrons are going to go to the oxygen. So this is going to be partially negative, partially positive, partially positive, partially negative. And partially positive and partially negative charges, they like each other, they attract to each other. But it's not strong enough quite to make a bond, but there is very, 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 very like strong attraction between them. They want to stay next to each other. So this is what forms that whole bond. And then in nucleotide bases, it's the same idea of the partial charges.
Yeah.
It's not always oxygen, it's like oxygen, nitrogen, and hydrogen that play over there, because nitrogen is also a highly electronegative element. But what I'm trying to get at is that so there's C and G in DNA, and then there's T and A. These form two bonds between each other. You just remember that. You don't need to like really understand it, but there's only two places in these where hydrogen bonds can be formed. And then C and G form three hydrogen bonds. So there's three places where electronegativity difference is large enough for there to be a partial charge and for them to come together in a hydrogen bonding manner. Essentially, this means that base pairing between C and G is stronger than base pairing between T and A. Okay, also, so in currently accepted theory of how life came to be on Earth, the initial information molecule was RNA. So even though right now most organisms alive use DNA, it was RNA that is assumed to be the first thing that was coding for information, and that is because RNA itself can function as a riboenzyme. So it can pretty much help translate mRNA into proteins. So well, sorry, no, that's about it.
No.
Let's just think over ribozyme. So ribozyme, it sounds like an enzyme, right? But instead of instead of N, it's a ribo. So ribozyme is an enzyme that's made out of RNA. Usually enzymes are proteins, which are made out of amino acids. But a ribozyme is same, a function, which is decreasing activation energy of reactions and catalyzing increasing speed of the reaction. But it's just made out of RNA and nucleotides that make up RNA. So these also are separated into two types. So nucleotide bases are separated into two types. There's purines and then there is pyrimidines. So purines, the main difference in structure between them is that pyrimidines are one 6-member ring. Purines is 2 rings, one 6-member, one 5-member rings bound to each other. That's just the difference. You don't need to know that. I'm just saying like they're different. Okay, and what you do need to remember is that purines it's A and G, and pyrimidines are C, U, T. U and T are obviously the same, just DNA, RNA. So all you really need to remember is C and T.
C and T.
And then as long as you remember that T gets switched for U, you remember that it's also a pyrimidine. How you're gonna remember this is pure as gold. Purine as gold. So pure as gold, and you know that PAG. So pure AG. So it is like, honestly, I think it's not easy to differentiate between purines and pyrimidines. Like when you're doing a mnemonic like this and you're relying on your like the way you pronounce it, but think the letter U, pure, because like some people can say this pyrimidines, right? So it's gonna sound the same. So if you're gonna use this, just make sure that you're clear on the pronunciation. I think that might be difficult. And then over here, you're gonna remember this as cut the pie. And like, I don't know, think wedding, like gold rings, pure as gold, and then what that wedding you also cut the pie. I don't know, like if that makes some sense for me. So in this case, it's CT pie, and then you remember that pyrimidines. Even though it is a Y right here, you're relying more on the pronunciation, the phonetics of it.
Yeah.
So, because there might be a question, which of these are pyrimidines, which are purines, and then you remember that. Another way to remember is, you know that C binds to G, T binds to A. So, if you definitely remember that T is a pyrimidine, you already know that A is not a pyrimidine because it has to be one pyrimidine binding to another purine. You know what I mean?
Yeah. You have to,
So like that's an exclusion that you like can keep in mind. Okay, so types of RNA in cells. mRNA, messenger RNA, rRNA, ribosomal RNA, tRNA, transfer RNA. Those are, I think, common. the one that is introduced in this class is snRNA, which is small nuclear RNAs, and they assist in RNA splicing. They also help regulate gene transcription, and so pretty much to my understanding what it does is RNA in our body, it doesn't like our body does not like RNA to be double-stranded. So DNA is double-stranded, RNA is single-stranded. The only thing that wants to be double-stranded is a double-stranded RNA are viruses. So there's a group of viruses that have double-stranded RNA. So I think this is one of the defense mechanisms that our body is able to employ against double-stranded RNA viruses. And as soon as it sees RNA that's double-stranded, it wants to destroy it. So these small nuclear RNAs, they're synthesized by our body for the specific purpose of finding another. So for example, like there's this long mRNA, and then that small nuclear RNA double binds to it right here, and then it can cut out this portion, or even the whole RNA can be degraded. So it kind of marks the mRNA for degradation by making it double-stranded. So pretty much just remember that small nuclear RNAs assist in RNA splicing and degradation. The four main kinds, one more time, mRNA, easy, protein translation, right? Messenger RNA. rRNA, ribosomal RNA, which is kind of like a riboenzyme. Yeah, is that what I said? Yeah, riboenzyme. Which ribosomes are made up out of it. And then transfer RNA, they hold amino acids and they bring amino acids to ribosomes during translation. tRNA is the most abundant type of RNA in the body. And small nuclear RNA assist in splicing and degradation of RNA. So eukaryotic transcription requires, like we said, transcription factors and enhancers. The classical example of this is the lactase gene, which is basically it generates the generation of the enzyme that breaks down lactose into galactose and glucose. So so this right here codes for that enzyme. This right here is the promoter region. And it has like a thing sitting right here that blocks RNA polymerase from binding and synthesizing this enzyme. When there is lactose, and this is the promoter region, and so when the lactose is present, it binds to here, this changes conformation, moves away, so the promoter region is active, is available for RNA polymerase, and then transcription factors are able to bind to it. So just this, it's not really complicated there's obviously multiple layers of complexity to this, but you just need to remember that we need transcription factors and enhancer elements for eukaryotic transcription. For prokaryotic transcription, not so much. It says right here, sigma factors assist RNA polymerase recognition. Okay, so what would be some effects on the cell if the function of a ribosome was inhibited? Ribosome. Like, what can you think of?
Well, ribosomes synthesize proteins.
Yeah.
So they wouldn't be able to
They won't be able to
move
complete
on.
the function.
Yeah.
Yeah. Yeah, exactly. So just, proteins are going to be coming out either
Defective.
short, yeah, or not coming out at all, and or maybe like too long or whatever. Basically, that's going to be affected. I see the cancel type. Okay, so this like question number 4 right now, in the survey, like you've done it in your class, right? But I think this is like a pretty good example. So a team is investigating why a certain cancer cell type has increased M6A RNA modification. They have hypothesized this modification enhances specific oncogene expression. The goal is to determine the functional consequences of increased M6A levels. So right now for me, I'm like, M6A, what is that? I don't remember seeing that in the slides. So like if I'm on the exam, like I forgot, I don't remember what M6A is. But now remembering the question, how does increased M6A modification most likely contribute to cancer progression? So A is increases. How does increased M6A modification most likely contribute to cancer progression? Increases mRNA decay, reducing oncogene expression. If we're reducing oncogene expression, we're not contributing to cancer progression. We're actually stopping cancer.
Right.
So even though like I don't know what M6A is, I can still eliminate that answer.
Right.
B enhances translation efficiency, increasing oncogene activity. That sounds promising.
Yeah.
C causes random mutation in RNA sequence. Okay, maybe. Okay, D reduces the ability of ribosomes to recognize mRNA. That would reduce expression of the protein. It would reduce expression of the gene. So again, D cannot be that.
Right.
So we're back to B and C again. So the question is, how does increased M6A modification most likely contribute to cancer progression?
Or it have to be B?
It would have to be B, yeah. And because it's a most likely contribution, so what's the most likely effect? And then they give you a specific RNA modification, M6A. So there's a specific thing. So it cannot be causes random mutation.
Right.
So yeah, enhances translation efficiency, increasing oncogene activity. Okay. We already went over this. The next slide is going about RNA processing. Please make sure you understand RNA processing by the next test, okay? It's 3 steps: intron splicing, cap and tail. Cut it up, put the cap, put the tail. Intron splicing, 5' methyl G cap, poly A tail. Then we're ready to be translated. Exons are left, introns are intruders, they're cut out, they're Abnormal splicing leads to non-functioning protein leading to disease. Translation occurs on ribosomes. mRNAs are scanned, and then tRNAs deliver amino acids to ribosomes, and peptide bonds form amino acids together into proteins. Then every amino acid has like a couple of codons that are specific for that amino acid, and then so we have 3 stop codons, which are you are gross, you are annoying, you go away, and then we only have 1 start codon, which is AUG, as in August. Fall semester starts in August. It's gonna be AUG, and so the stop codons, they do not code for any amino acids. This does code for an amino acid methionine, or it's abbreviated as MET, or just the letter M. So human proteins start with methionine. And then ribosomes are like made up of multiple subunits. They call them the small and the large. Honestly, I'm gonna move past this because I do not remember being quizzed on this at all by Dr. Okay? I wanna really stay focused on things that applicable to the grade with strategies. Okay, so then there's we can also modify things post translationally. So if we are modifying anything post translationally, it means that translation has already been completed. So if it's post translation, we're talking about modification of proteins. Post transcription is modification of mRNA. So after it was transcribed, it can still be modified. Post translation is modification of proteins. What it's referring to for the most part is to phosphorylation, methylation, and ubiquitination. So phosphorylation, I think, is the most common thing that people in biology classes start talking about early, and that's just transfer of a phosphate. And remember when we were talking about the DNA backbone, it's phosphate sugar. So literally that same phosphate, the phosphate that has 4 oxygens, like this. So over here, electrocounts, the one that Dr. Melikian does. If you were to do it for this oxygen, it would be 1, 2, 3, 4, 5, 6 over 8. Oxygen wants to be 6 over 8. So this one is neutral. This one is neutral. 1, 2, 3, 4, 5, 6, 7. 7 over 8. This one is -1. This one is -1. So this is why people say the phosphates are negative, and then this negative charge is the reason with this extra pair right here is gonna find something and bind to it. So with this, I'm just trying to show you that like concepts in biology, you can draw parallels. And so attachment of this phosphate to a protein, it's gonna give it a negative charge, which is gonna cause change in conformation, or something's gonna change because of that negative charge, which is gonna be modification post-translation, because we already translated it fully. The protein is not going to be modified, it just gets an additional thing attached. Then there's methylation. And methylation, if you remember, we were talking about methylation and acetylation of DNA. So what does methylation do? What does acetylation do?
Methylation, it condenses and it stops from the
Exactly.
entering.
Yes.
And acetylation, it allows for
Correct.
entry.
Exactly. That's all you need to remember. Do not overthink it. Just remember that on the test, it can be described as deacetylation. Deacetylation is removal of acetyl group.
Okay.
So that's also kind of condensing it. You know,
Mm.
if he wants to be, that's just thinking a little bit more in depth. If he wants to,
Yeah.
so don't automatically assume that if you see the word acetylation,
Okay.
make sure that it's, there's no tricks over there. And then ubiquitination. So ubiquitin has a whole bunch of roles in the body, but if a protein is tagged with ubiquitin, it's either going to be expelled outside of the cell, so for example, like released into the bloodstream or released into the GI tract, or it's going to be sent to the lysosome for degradation. So basically, ubiquitination means like get out of here. Like if something is ubiquitinated, like the cell wants it to be gone.
Oh, okay.
Lysosome degrade, ubiquitinated proteins misregulated to degradation can lead to neurodegenerative disease. So also another type of mRNAs is microRNAs. They're abbreviated as miRNAs. And I mean, it's similar to small nuclear RNAs. They regulate gene expression. They bind to RNA, making it double-stranded, and then so that the body wants to degrade it. So this is like one of the ways that the body can control protein expression. So for example, if I'm under a lot of stress and I need like a certain and the body can't really afford to synthesize a certain protein. It needs to allocate its resources to a different place. So instead of just stopping transcription, which is, it would make sense. Sometimes what the body chooses is it keeps transcription active, but it doesn't let it go to translation, which is it's a less efficient way to control, but depending on the scenario, it actually might be better in some cases. Okay. And so the thing that prevents in these cases mRNA from going to mRNA to being translated is these miRNAs. So think of miRNAs and snRNAs as similar in function.
Okay.
As like controlling RNA expression and things like that. Okay, what is a silent mutation?
Well, it's like the way it says silent, it comes in and mutates a protein.
So a silent mutation is a change in nucleotide base that does not change the amino acid. Okay. Amino acids have, most of them have 3 codons that are able to code for it. So pretty much, like some of them have 4. Some of them have 2. Yeah, it's either 2 or 4 actually. It's not. Some of them have 3 too. Okay, so basically they have a couple, right?
Mm-hmm.
And so over here, so the amino acid is serine, and then it's UCU, UCC, UCA, and UCG. Okay, so you see that these two
Mm-hmm.
are all the same.
Yeah.
The third one is the one that
They
changes.
change.
So this was, I think this was in my evolutionary biology class, but just so you have an understanding, this one is like the least important one. This is like the second important one, and this is the most important one.
Okay.
Because like if you change this one, most likely it's not going to be a cell mutation. But what I'm trying to get at is, look, UCU, if this gets mutated to C,
Mm-hmm.
it is a mutation. But we have the same amino acid, so the primary structure of the protein at the end is going to be the same, so there's no effect.
Okay.
In reality, there is a little bit of effect,
Mm-hmm.
because like the speed at which this gets translated and this is a little bit different, so but essentially there's no effect. So it's a silent mutation. It is a mutation. U went to C or C went to A or whatever it is, but there was no change in the primary structure.
Got it.
Then non-silent mutation is a mutation that causes a change in amino acid. And then there's two types of non-silent mutation. There's a missense mutation,
Mm-hmm.
and then there's a nonsense mutation. So think nonsense, like it's nonsense.
Mhm.
So the protein that's finished, it's like completely, it doesn't make any sense at all. So
Right.
basically nonsense mutation is a premature stop codon. So if, for example, we were coding for tyrosine, which
Mhm.
is AUU, and then this U gets mutated to A, so you are annoying. That's a stop codon. So instead of translating tyrosine and then translating 100 more amino acids,
Mhm.
it's just gonna stop and be cut off. So again, completely non-functional protein, nonsense mutation. This is the worst kind of mutation. It's just like you can't do anything about it.
Okay.
But if instead of which one? UA. Oh, C. Over here. So we have phenylalanine BUUU, and then we have leucine BUUA. So we switching phenylalanine to leucine,
Mm-hmm.
mutation of one right here, which is different amino acids,
Mhm.
but it's not a stop codon. So it is a non-cellular mutation, but you're still gonna get the same protein. The only difference is gonna be this one amino acid. And you know, when it's like 1,000 amino acid long protein, one amino acid is most likely not gonna be the end of the world. You are still gonna have a somewhat functional protein,
Mhm.
but in some cases, yeah, it can be pretty devastating if it's like an important amino acid. Like for
Right.
example, if you can't phosphorylate a protein anymore, so this gets bound to like one of the amino acids. There's two amino acids that they usually binds to, and if it can't do that, then yeah, that's devastating. But if it's like a simple structural thing, then it might be not so bad. So it's considered a missense mutation.
Okay.
So nonsense, like really
Makes
bad.
no
It doesn't make any
sense.
sense at all.
Yeah.
Missense, it's might not be so bad. Silent, we cannot tell. since we are on this mutation type of thing, I wanted to just say another word about this. So there's can also be addition and deletion mutations, right? So that's self-explanatory. Addition is we are adding an extra nucleotide. Deletion, we are deleting some portion of it. So these type of mutations can lead to frame shift. So ribosome moves in threes, right? So this is one amino acid, this one amino acid, amino acid, amino acid. And if you add or delete nucleotides that are not divisible by 3, so if 1, 2, 4, 5, etc., you're gonna have a frame shift, which is also really devastating. What I mean by this is, look, so right here, normally it's this amino acid, this amino acid, this amino acid, this amino acid. Now we put just one nucleotide base here, it's still this, but now it's gonna be this, this, this. So it's like
changing.
It shifts everything. So it doesn't only affect this codon. It affects everything that goes afterwards. So all of these are going to be changed, and there is a high. All the amino acids are going to be different. There's also a high chance of there being a stop codon. So a frame shift mutation is also pretty devastating, and it happens when there's addition or deletion in of nucleotide bases that's like not in intervals of 3. Because if we were to, for example, add 3 things right here, it would actually be less of an effect, because all we're gonna do is just add an extra amino acid but keep the sequence the same. And adding one amino acid might not be so detrimental when compared to altering literally everything else. The same goes for if we just get rid of one amino acid instead of get rid of 3 nucleotide bases. Instead of just getting rid of one, we might just lose one amino acid, and if it wasn't a really important one, the protein might still be somewhat functioning. Okay, girl. It's 9:30. Do you have any questions?
No.